Hello, today we are going to see the excretory system of Hirudinaria, that is leech. The basic unit of excretory system of leech is nephridia. The excretory system of leech consists of 17 pairs of nephridia. They are arranged from 6th to 22nd segments, one pair in each segment. The nephridia are of two types, testicular and pre-testicular. The last 11 pairs of nephridia which show contact with testis sac are called as testicular nephridia, whereas the first 6 pairs which do not show contact with testis sac are called as pretesticular nephridia. First we will see testicular nephridia. These are last 11 pairs of nephridia present from 12th to 22nd segments, showing contact with testis sac. Hence, these are called as testicular nephridia. Now we will see structure of testicular nephridia. Now this is the complex structure of testicular nephridia and body wall showing nephridiopore. A typical testicular nephridium is a horseshoe shaped structure. Each nephridium consists of the following 6 parts. First, main lobe. This is main lobe. Second, vesicle and vesicle duct. This is vesicle and vesicle duct. Third, apical lobe. This is apical lobe. Fourth, inner lobe. This is inner lobe. Fifth, initial lobe. This is initial lobe. And sixth, ciliated organ. Here you can see ciliated organ. Now we will see each part of nephridium one by one. First, the main lobe. It forms the major part of the nephridium. It looks like a horseshoe. It is ventrolateral in position and lies in between two adjacent ceca of the crop. It consists of two unequal limbs, anterior and posterior limbs. The anterior limb is longer and posterior limb is shorter. Cells of main lobe are big and polyhedral in shape. The second part is vesicle and vesicle duct. A narrow vesicle duct arises from the ventral end of longer anterior limb of main lobe and runs posteriorly to open into a large thin walled bladder called terminal vesicle. It is situated ventrolaterally behind the rest of nephridium. The vesicle is large oval sac bounded with a non-contractile thin wall. It opens outside through a short and narrow excretory duct called as nephridiopore, which we have seen in figure 1. The sphincter muscles are located at the junction of vesicle and excretory duct. These muscles regulate the outflow of excretory substances. The vesicle is non-contractile and its internal epithelium is non-ciliated. Third is apical lobe. The posterior limb of the main lobe continues to form a stout apical lobe. It lies in an anterior-posterior position beneath the crop. Its anterior end is slightly swollen and bent on itself like the handle of a walking stick. The cells of apical lobe are big. Fourth is inner or recurrent lobe. It lies in the concavity of the main lobe and also runs forward along the outer side of the apical lobe for about half of its length. Fifth is initial lobe. This lobe is very long, narrow cord-like structure. It is composed of single row of elongated hollow cells. It remains coiled around apical lobe. Its posterior end joins the main lobe while its anterior free end runs inward up to the testis sac and blindly ends near the perinephrostomial ampulla. Sixth is ciliated organ. This is a structure of ciliated organ and single funnel. Ciliated organ lies inside the perinephrostomial ampullae. It remains suspended from the inner walls of the ampullae by 4 or 5 strands or trabeculae. It is a modified and compound structure. It consists of a spongy central reservoir and peripheral ciliated funnels. The reservoir is a perforated spongy chamber. It contains a central mass of connective tissue cells and an outer wall of a single layer of cells. The wall contains numerous pores. Each ciliated funnel is like an earlobe, having a ciliated broad distal free end and a narrow proximal neck, which fits into a pore situated on the wall of reservoir. In the embryo, ciliated organ has a distinct cellular connection with the nephridium. But in adult, it loses its connection and excretory function and becomes a part of the hemocoelomic system by producing coelomic corpuscles. So far, we have seen about the various parts of testicular nephridium like the horseshoe-shaped main lobe, the bladder-like vesicle, a narrow vesicle duct, the inner lobe in the concavity of the main lobe, the walking stick-like apical lobe, and a cord-like structure that is the initial lobe which ends in the testis sac, and the ciliated organ and the perinephrostomial ampullae. Now we will see about pre-testicular nephridia. First 6 pairs of nephridia, situated in 6 to 11 segments, are called as pre-testicular nephridia because these segments do not contain testis sac. There are no perinephrostomial ampullae and ciliated organs. The initial lobe of nephridia terminates into loose connective tissue of body wall instead of testis sac. Except these features, rest of the structure of pretesticular nephridia is same as testicular nephridia. Now we will see about histology of nephridium. The lobes of nephridium are formed of mass of gland cells. Gland cells contain minute intracellular ductules, which open into a wider intercellular central canal. The central canal leaves the main lobe as vesicle duct. A nephridium is richly supplied with hemocoelomic capillaries. Now we will see about the physiology of excretion. Nephridium is both osmoregulatory and excretory in function. Excess of water and nitrogenous wastes are excreted out by the nephridium. Nephridial gland cells separate the waste products, chiefly ammonia, from the hemocoelomic fluid. The excretory fluid is finally collected in the vesicle and excreted out through the nephridiopore. Leech excretes hypoosmotic urine. The salts are reabsorbed from the urine by the nephridial tissues. Here we have finished about the excretory system of Leech. Thank you.